Archaeoglobus is a genus of the phylum Euryarchaeota. Archaeoglobus can be found in high-temperature oil fields where they may contribute to oil field souring. Metabolism. Archaeoglobus grow anaerobically at extremely high temperatures between 60 and 95 degrees Celsius with optimal growth at 83 degrees Celsius. They are sulfate-reducing archaea, coupling the reduction of sulfate to sulfide with the oxidation of many different organic carbon sources, including complex polymers. A. Lithotrophicus live chemolitho autotrophically from hydrogen, sulfate and carbon dioxide. Also A. profundus grow lithotrophically, but while this species needs acetate and CO2 for biosynthesis, they are heterotroph. The complete A. fulgidus genome sequence revealed the presence of a nearly complete set of genes for methanogenesis. The function of these genes in A. fulgidus remains unknown, while the lack of the enzyme methylcom reductase does not allow for methanogenesis to occur by a mechanism similar to that found in other methanogens. Description and significance. Archaeoglobus members are hyperthermophiles that can be found in hydrothermal vents, oil deposits, and hot springs. They can produce biofilm when subjected to environmental stresses such as extreme pH or temperature, high concentrations of metal, or the addition of antibiotics, xenobiotics, or oxygen. These archaeons are known to cause the corrosion of iron and steel in oil and gas processing systems by producing iron sulfide. The biofilms, however, may have industrial or research applications in the form of detoxifying metal-contaminated samples to gather metals in an economically recoverable form. Genome structure. The Archaeoglobus fulgidus genome is a circular chromosome roughly half the size of E. coli at 2,178,000 base pairs. A quarter of the genome encodes preserved proteins whose functions are not yet determined, but are expressed in other archaeons such as Methanococcus janusii. Another quarter encodes proteins unique to the archaeal domain. One observation about the genome is that there are many gene duplications and the duplicated proteins are not identical. This suggests metabolic differentiation specifically with respect to the decomposing and recycling carbon pathways through scavenged fatty acids. The duplicated genes also gives the genome a larger genome size than its fellow archaeon M. janusii. It is also noted that Archaeoglobus contained no inteins encoding regions where M. janusii had 18 molecular signatures showing relatedness to methanogens and thermococci. Comparative genomic studies on archaeal genomes provide evidence that members of the genus Archaeoglobus are the closest relatives of methanogenic archaea. This is supported by the presence of 10 conserved signature proteins that are uniquely found in all methanogens and Archaeoglobus. Additionally, 18 proteins which are uniquely found in members of Thermococci, Archaeoglobus and methanogens have been identified, suggesting that these three groups of archaea may have shared a common relative exclusive of other archaea. However, the possibility that the shared presence of these signature proteins in these archaeal lineages is due to lateral gene transfer cannot be excluded. Ecology. Archaeoglobus species utilize their environment by acting as scavengers with many potential carbon sources. They can obtain carbon from fatty acids, the degradation of amino acids, aldehydes, organic acids, and possibly CO as well. High temperatures are ideal growth temperatures for Archaeoglobus, although a biofilm environment provides some environmental elasticity. Biofilm is composed of polysaccharides, proteins, and metals. Medicine. Cells protected by biofilm are difficult to destroy using conventional antimicrobial therapy, which gives them medicinal possibilities. Scientific databases. PubMed references for Archaeoglobus. PubMed Central References for Archaeoglobus. Google Scholar References for Archaeoglobus.